Hey guys, welcome back to my YouTube channel. Today we are going to discuss another important topic from MHC or immunology, that is class 1 MHC molecule. I have drawn a rough structure over here. If you want to understand, we'll discuss the detailed structure later on. But to give an overview, I'll say there are three alpha domains. There's a single alpha chain which forms three domains: alpha 1, alpha 2, and alpha 3, and non-covalently attached beta 2 domain, followed by a transmembrane domain and a cytoplasmic carboxy terminal domain. Okay, and they alpha 1 and alpha 2 forms an antigen binding site where the antigen binds, and they bind on the surface of nucleated cell to present the binded antigen. Okay, now if it is clear to you till here, then it's okay. If not, we'll discuss, then you'll get to understand. Okay, let's start. What is class 1 MHC molecule and what is its structure basically? Organized into three main locus we discussed in the video earlier: ABC. These genes encodes glycoprotein expressed on the surface of all nucleated cells. This is a very important point to remember, that these genes encode protein which are expressed on the surface of nucleated cells. Class 2 is expressed on the surface of dendritic cells, and class 1 is on nucleated cells. Please remember this because it will be of further use also as we'll study endogenous and exogenous pathway. Okay, major function. Now let's discuss the major function. It is to present the peptide antigen to the TC cells. TC, cytotoxic T cells. Okay, consists of a large alpha chain. I told you, large alpha chain, which is of molecular weight nearly 45 kilodalton, associated with a non-MHC molecule called the beta 2 microglobulin, whose molecular weight is 12 kilodalton. Beta 2 encoded by gene on chromosome number 15. This is a point to be remembered. Earlier we discussed that uh, MHC genes are present on chromosome number 6, right? But here we can see beta 2 is encoded by a gene on chromosome number 15 because it is a non-MHC molecule. Okay. Alpha chain is a transmembrane. What is a transmembrane? A protein which travels or gets repeatedly transfers through the two membrane two membranes of a particular organelle or a structure or anything which passes the membrane again and again. Okay. Alpha chain is a transmembrane glycoprotein encoded by any one of the gene in ABC. ABC is the part of MHC1 complex. I told you, of HLA complex. Okay. Association with beta 2 microglobulin is necessary for the expression of class 1 MHC molecule on cell surface. This is very important thing, that expression beta 2 microglobulin is very necessary for the expression of class 1 MHC molecule. If it is absent, then it may result into non-expression or a disease called Dowdy tumor. Okay. Alpha chain is folded and organized into three extracellular domains. I showed you in the structure something like this, है ना? Alpha 1, alpha 2, and alpha 3. Each domain consists of approximately 90 amino acids, like 90, 90, and 90. So nearly alpha chain is composed of 270 amino acids. Nearly. It's not the exact value, nearly. These are followed by a transmembrane domain of 25 hydrophobic amino acids and a cytoplasmic carboxy terminal domain of 30 amino acids. Beta 2 microglobulin, only one extracellular domain similar to alpha 3 domain. I told you that this is the structure, suppose. So there beta 2 was organized, alpha 1 and alpha 2 had similar structures, and alpha 3 had a similar structure with beta 2. That is why both of them show association with each other. Okay, domain in structure, that is why it interacts extensively by non-covalent interactions. Alpha 1 and alpha 2 interact with each other at the surface of cell to form a deep cleft like ABS. What is ABS? Antigen binding site. I showed you this is the antigen binding site. Now let's discuss about ABS. ABS means antigen binding site. The floor of antigen binding site is made up of beta plated sheets formed by 8 anti-parallel strands. Something, suppose this is the cleft, so it has, like, if we view this part, it is like this. Okay, okay. Walls. Anti-parallel means one goes this side, one goes this side, right? Walls are made up of by alpha helical coils. This region is made up of alpha helical coils. It is a deep groove with dimension 25 Armstrong into 11 Armstrong into 11 Armstrong. It is large enough to bind peptide of size 8 to 10 amino acid. This is very interesting and important point that overall structure of class 1 molecule has a resemblance to the immunoglobulin molecule. How? The upper part, that is the alpha 1 and alpha 2, this part comparable to the antigen binding variable region because the antigen binds here, right? In antibody structure also, like see, we see this one, the antigen binds here, in same way antigen binds here. Okay, and alpha 3 and beta 2 forms the Ig constant region, like this region. This forms the constant region, right? Oh, sorry. Molecule is susceptible to papain cleavage. I hope you know papain cleavage. What it is means, papain means it cuts the molecule like this and this. See, this is, sorry, this is the antibody structure, right? So it separates out, it cuts in it into three parts. This, then, and cuts from this also and this also, right? In absence of beta 2 microglobulin, I told you this point is very important. Now see how. In absence of beta 2 microglobulin, MHC1 molecules are not expressed on cell surface seen in Dowdy's tumor cells, which synthesize MHC1 molecule but unable to produce beta 2 microglobulin. Hence, it has a non-functional MHC1 molecule, right? I have again repeated this structure, so hence it becomes comparable. This is the alpha 1 domain. Here this is the alpha 1 domain, right? This is the alpha 2, alpha 2. They form this, wait, I'll show you this way. They form this antigen binding site, and here it forms this antigen binding cleft, right? This is alpha, oh, sorry. This is alpha 3 domain. Wait. This is alpha 3 domain here, and this is alpha 3 domain. This is beta 2 microglobulin, and here we have beta 2 microglobulin. They show non-covalent interaction. And I told you about antiparallel strands. These are antiparallel strands which make the floor of antigen binding site. Here you can see this detailed structure. Alpha 1 domain, alpha 2 domain, alpha 3 domain. This is the papain cleavage site. It shows 13 amino acids. Okay, cuts up to 13 amino acids. This is the transmembrane domain, and this is cytoplasmic tail. I hope the topic is clear to you. If you have any questions, please drop down in the comment section. Please like, share, and subscribe to my channel.